Yeah, I am Dipanjan Ghosh. Here I present the RH factor, that is the Einsteiner winner. How do they observe the antigen, the antigen to the winner, to the rhesus monkey, and also it is present to the human body also. And here this is the RH antigen, and we know this is a potent antigen, and we see here basically the RH genetics. Basically, we know about that RH genetics, and basically how do the Fisher days and winner and the different types of nomenclature process, the CD technology and different technologies used just to different nomenclature processes used to determine. So this is the pattern, the arrangement, and this is the winner nomenclature. And basically if we go through that concepts, then we can realize. Here are basically different types of genotypes and versus phenotype has been shown here. Basically, this is the genotype, capital small cd. Here we have seen different types of arrangement. And why is RH incompatible? We know basically at the first during the first baby, the RH positive blood from fetus go to the mother's body, and it will generate the anti-RH, and during the second baby, that anti-RH go to the fetus, second fetus body, and it will generate, it will make the reaction RH and anti-RH. This is called the antibody dependent cell-mediated cytotoxicity. So this is, and that's why we know basically the hemolytic disease of newborn or erythrocytosis fetus actually occur. And this is the different types of sexual reproduction process, that is different syngamy process, isogamy and syngamy. And here the sexual reproduction of fungus, how do they happen? And this is the basically the quasar one I mentioned. Quasar is basically formed by carbohydrate protein lipid. It's the combinations of carbohydrate protein lipid, whereas the microsphere is the only the protein. So you have to keep remember what is the basic difference between microsphere and quasar whenever we define. Microsphere is basically the gathering of amino acid, whereas quasar is made up of basically lipid, protein and carbohydrate. And this is the ABO blood group proposed by Einsteiner, and how do the AB and O is actually generated on the basis of the AB antigen to the RBC and the alpha beta antibody to the serum. And this is the blood coagulation process. We know there are different coagulating factors, and this is the RBC precursor structure. How do the glucose, N-acetyl glucosamine formations of H antigen? How do the H antigen we convert to A antigen and B antigen? In case of Bombay blood group, H antigen remains absent. And this is the pictorial presentation. How do the blood